Hello everybody, thanks for joining to our new webinar. Today we will talk about the importance of choosing the appropriate disposable when assessing semen samples. So the first thing that we need to take into account is just to ask ourselves what type of disposables may I use for doing its test, which are the difference between the different disposables for doing the same test, if are my current results accurate and reliable, or if there is any way to improve the efficiency of the procedures that I am doing in my routine. So let's start with the first point. Here in that slide I have shown that there are lots of different disposables available in the market for doing the same test. Okay, so for instance, there are lots of different staining solutions for evaluating the sperm morphology, or different counting chambers for assessing the sperm concentration or the sperm motility. Okay, so what are the difference between this type of disposable? So when we are starting evaluating a seminal sample, a semen sample, we need to evaluate first of all the seminal plasma features, because this will provide from the background for the environment that will help us to do a good interpretation of the results. But what happens when we are working with pathological samples in which there are not clear liquefaction, the liquefaction process is not complete, or maybe when there are very viscous samples that are very difficult to handle. In that cases, in the market, you will find different profiles of disposables that could solve the problems. We have distinguished two types of profiles, like could be the chemical enzymatic disposables from the mechanical disposables. According to WHO, the enzymatic reagents could be used, like be the double-cause phosphate buffer, or maybe the addition of bromelain could be incorporated with the semen sample in order to break the coagulum that is present when liquefaction is not complete. Okay. So this could solve our problem, but it could entail some problems on the sperm features, since could affect on the biochemistry of the seminal plasma. It could affect on the expected motility for that sample on natural conditions, or maybe could alter the original morphology of the sperm cells. Okay, this is why it is recommended to use mechanically methods for breaking the mucus strands that is compacting all the sperm cells and is making difficult the handling of that samples. An example for that type of that profile of reagents are the viscoflux. Here, this is an example of the viscoflux kit provided by MICROPTIC that consists in a syringe with a specific needle that will help us to break mechanically these mucous strands from the seminal plasma, okay, without affecting any sperm parameters or sperm features. Okay, so this could be a good solution for handling that type of problems. On the same way that we are evaluating this viscosity or this type of features of the seminal plasma, it is important to take into account the biochemical test, especially the pH assessment that will provide from very valuable information about the functionality of the accessory glands. For evaluating the pH, you can easily use a conventional pH strip, or if you are using a CASA system, we have designed this type of counting chambers that at the same time that allows to evaluate motility and concentration. Also there is a pH strip that allows to do simultaneously the pH lecture. You can do it manually or automatically. Okay, so here there is the lecture in the protocol, so it is not necessary to do it automatically. You can also do it on a manual way. Also could be interesting to complete your analysis about the seminal plasma by evaluating the levels of citric acid within the ejaculate. This is very important because this is a marker of the prostate functionality. Hence, biochemical tests like this one, like the citric screen. There is a colorimetric kit that contains this plate that will quantify colorimetrically the quantity of citric acid on its ejaculate. Okay, so this could be very useful. In case in which we are observing the sample at the microscope and we detect a lot of agglutinates, lots of sperms that are attached to each other but they're still moving, in that case could be interesting to also perform some immunological tests, like the MAR screen test. You can find this type of kits for different antibodies. Normally it is assessed the immunoglobulin A or G, and this type of kits are composed by two solutions. There is one solution that contains some bits that are coated with antibodies and antiserum. Then when mixed with the semen sample, allows the binding of the sperm cells to the different bits. Hence you can count how many sperms have antibodies and bits attached to these antibodies. On the same way that we are evaluating the semen parameters, as soon as possible it is very important to assess for the sperm motility because it's very sensitive to the pass of time. Here it's very important to select the best disposable for doing the analysis. It should be very accurate and reliable. Okay, so in the market you can find two profiles of disposable. This reusable disposable, reusable counting chambers, that could be the Neubauer, the Thoma chamber, Mackler chamber, and the disposable chambers. The reusable are very popular because you can use it several times, but it entails a very huge problem. There is the risk of contamination. If these are not handled properly, or if they are not cleaned properly, there is a high risk of contamination between samples and between the sample and the technician that is handling that sample. Okay, so also there are some, depending of the type of counting chamber that you select, you need to dilute a lot the sample in order to do the counting, and this could introduce a small measurement error on your results that could affect on the reliability of results. Or in some cases, when you put the drop on this type of slides, this sample is not spread homogeneously. Therefore, the results obtained are not very accurate. This is why more frequently the laboratories are incorporating the disposable chambers, like this one, okay, like this one that I show you. Or maybe if you are doing multiple analyses, you can also incorporate multi-chambers, okay, that minimize the contamination between patients, okay, and the results are very accurate and reliable since the sample is spread by capillarity and distribution of cells is fully homogeneous all over the chamber. Also, I would like to talk about the false counting chamber, because one of the most economic ways to evaluate motility and concentration is just using slide and a cover slide. In that case, we need to take into account that this is the less accurate way to assess motility and concentration. Okay? Why? Because we cannot control the depth of the chamber once we are putting the cover slip. Also, the distribution of cells is not homogeneous at all, and we cannot control the drifting effect on the periphery of the cover slide. Okay? Also, it's very common to find so many bubbles that could disturb your results. Therefore, despite of being used for so many laboratories none of the accredited laboratories are using this type of chambers because these are not accurate at all. Apart from the type of counting chamber that you prefer to use for your standard analysis, it is important to think if the method used for doing the assessment is the most accurate. For instance, when there are pathological samples with a very low sperm number, with lots of debris in the background, or maybe when you are using a sophisticated extenders that could introduce some noise in background, in that case, it could be worthy to work under fluorescence methods. Hence, there are some kits available in the market that could be the FluoCount, this one, okay, that contains a specific DNA label for highlighting these cells that are present in the ejaculate and omit all the debris, all the noisy background that could interfere on your analysis. This is especially recommended when you want to analyze vasectomies, oligospermia, or animal samples with a lot of debris. On the same way that we need to think about the way, the method for assessing the sample, also is important to take in mind the temperature that we will take into account for doing the assessment. Because it's not the same, the motility features, the sperm behavior will not be the same at 21 degrees than at 25 degrees than at 35 degrees. So we need to simulate as much as possible the natural conditions of the sperm cells on the environment where the fertilization occurs. To do that, we can just incorporate some cooling stages, some heating stages that modify the room temperature to adjust to the real conditions, so the natural conditions where fertilization occurs. Okay, here there is one example of an USB heating stage. You can put it easily in the stage of the microscope and connect it to the computer, and hence you guarantee that on all surface of the slide the temperature is at 37 degrees. Conversely, if you are working with a fish species or marine invertebrates in which the temperature should be below this body temperature, you need to work at 10, 15 degrees, you need a cooling stage. Again, you can put it on the stage of the microscope stage in order to preserve the best conditions for assessing sperm motility under natural environments. Now we will move to morphology tests. Here, it's very common to find a lot of different staining solutions, lot of reagents that will help us to evaluate sperm morphology. But which is the best? What we should to take into account for selecting one or another? The first important thing to take in mind, it is just if the coloration select is staining all the areas that we want to analyze in the sperm cell. That normally there are the acrosome, postacrosome, midpiece, tail, cytoplasmic droplets, and vacuoles. Okay. After doing a first list of all these solutions that allow to label all these areas, it's important to take in mind that these solutions are not affecting the original morphology or morphometry from our cells. This is why it's important to control the osmolarity for these solutions. We have to think that when we are staining one sperm cell with a hyposmolar solution, by natural process, by osmotical process, the heads tend to swell. Okay, this is what normally happens when we are staining with a deep quick-like solution. Conversely, when we are using hyperosmolar solutions, the process is the opposite. The sperm cell loses their water content and therefore shrinks the heads. This is what could happen when we are staining with Papa Nicolau or short colorations. Okay, so this is why MICROPTIC has developed the sperm blue coloration that is the unique isosmotic solution available in the market. It is allowing the labeling of all the areas distinguished in the sperm cell without affecting any morphological, any morphometrical parameter within the sperm cells, and also is compatible with multiple species. Okay, this is not only useful for human, it's also useful for animals. Okay, but for these laboratories more focused in human pathology, maybe they are also interested in staining other cells that are not sperm cells, that could be the round cells. In that cases, there is also the possibility to use a Diffquick-like stain, like could be the sperm stain ready to use. There is a most cost-effective solution that allows to do a specific staining for the sperm cells, but also for the round cells. Okay. On the same way that we need to think about all these issues about staining solutions, especially when you want to be accredited, it is important to standardize as much as possible your procedure for performing morphology. Okay? In that case, it's very important to standardize the smearing, the preparation of the samples for evaluating morphology. Now, this is possible thanks to this type of machines, okay, in which we will just put 2 slides here. We will put 10 microliters drop of sample on each slide, and then we will just press the lever. Here, this machine automatically is spreading the sample following the WHO indication. Okay, so this is a very, very nice option for standardize this procedure and be accredited. Also, if you are not able to use the liquid solution for evaluating morphology, you can see that in the market there are also available some pre-stained slides. These pre-stained slides, there are some points that we need to take into consideration because some of them normally are in the procedures indicate to put 1 drop of the sample and put the cover and analyze at the microscope. Okay, you have to think that when you put the drop and the cover, so many sperm cells will be located on a lateral position. Therefore, after evaluating at the microscope, you can confuse, you can erroneously classify one thin, one tapered sperm cell that is normal. Okay, this happens because of the position of the sperm cell, not because there is a real abnormality. So this is why it's important to, instead of using pre-stained slides, just to do the smearing on the same way that if you were using a conventional slide, in order to guarantee the optimal position of the sperm cells and then optimal classification of the different abnormalities. These pre-stained slides could offer some advantages for these laboratories in which there is not allowed the entrance of liquids inside because of problems of toxicity, and also could be very useful for these centers that need to work in the field, and therefore they cannot bring so many solutions. Okay, they are very easy to use. But here there is an example of our pre-stained slides with sperm blue. Okay, these are the morphology slides that are pre-stained with sperm blue. Okay, that provides from very, very good results after staining. And this is something very important to take in mind, because in the market you will find a lot of pre-stained slides that doesn't provide from the same accuracy than the liquid solutions. Conversely, with this type of slides, here I have shown 2 pictures. of same preparation. One's performed with liquid sperm blue, the other performed by using these pre-stained slides. Okay, so here you can see that there are not so many differences between them because the detail, all the quality of the image is exactly similar between liquid and this pre-stained slide. This is something we need to take into account because so many slides doesn't provide from the same accuracy, from the same details than liquid solutions. Okay. Another point to take into account is just to decide if it's worth it to wash or not wash samples for evaluating morphology. When to do it? We can do it when there are viscous samples in which the seminal plasma is introducing some noisy background, when the sperm concentration is not optimal for doing a fast analysis of the sample, or when there are lots of debris, or you are using extenders that are very viscous and are interfering on the visualization of the sperm cells. Okay, so you can use lots of conventional sperm washing methods, sperm washing medias that are available in the market, but in some cases these are a little expensive. So for some laboratories it's not worth it to use that type of solutions. This is why from MICROPTIC we recommend using the PBS, okay? The PBS, that is a saline buffer with the optimal composition for not inducing any morphology change on the original structure of the sperm cells. These are provided on this type of pellets that can be stored for long period of times at room temperature, doesn't require from specific storing conditions, and you can dilute each tablet with 200 ml of deionized water and use it for simple washing. Okay, you can just centrifuge the sample and replace the seminal plasma by PBS. On that way you will minimize all this noisy background that could interfere, that could overstain our semen sample. Okay, but if you are evaluating the round cells, in that case it is not recommended washing samples. It is better just to preserve one replicate performed with a semen smear in order to evaluate the morphology of round cells and which is the ratio between the cells and the round cells, okay, in order to get realistic results. Also, another decision to think about is just if it's worth it to mount or not mount preparations. Once the smears are stained, you have the option to immediately analyze under the microscope or just to mount and then analyze. We have to think that the sperm cells are three-dimensional structures. Okay, so what does it mean? That when you are working at high magnification, depending on the focus plan that you are working on, you will see some structures or not. When you are mounting, optically, all these structures become more flattened and everything is going to the same focus plan. Hence, it is easy to focus and evaluate all the structures on the same focus plan and make the analysis easier and faster. Okay, you can use EOKit or similar mounting media, synthetic mediums, but these are especially important when you are using gas assistant because they are very accurate methods for measuring sperm morphology, and the detection, the sensitivity to detect the different structures is very different depending if you are analyzing mounted or not mounted slides. Okay, so this is something to discuss about. Now we will move to the tests that are available for assessing DNA fragmentation. You will see that so many laboratories are using flow cytometry, but this is a very expensive and a very complex method for evaluating DNA fragmentation. So just to mention that there are other protocols available, that could be the tunnel assay, COMET, or the sperm chromatin dispersion test. Once we are just evaluating which is the best technique to use for evaluating DNA integrity, It's important to think about on the disposables that are required for doing its sample preparation. So, for instance, for doing tunnel aside, that there is a very accurate method for evaluating DNA, we have to think that there are some reagents that require the use of some reagents that are very cancerogen, therefore are dangerous for the health and not recommended for some laboratories. Okay. On the same way, for doing the comet assay, you will need to use the electrophoretic gel. You will need to use the electrophoretic machine. That is not easy to find in andrology laboratories. This is why it is recommended to use the sperm chromatin dispersion test, like this one. Okay, there are so many available in the market. With that kit that we provide, there are available all the reagents required for doing the sample preparation, okay, including the staining solutions, and provide some very valuable information about the different levels of DNA fragmentation within a semen sample, and allows a good repeatability of results in the laboratory. So this is one of the most recommended techniques for evaluating DNA integrity. On the same way, for some laboratories, it is for special interest to do a first screening about the presence of reactive oxygen species. Okay? When there is a risk of oxidative stress, this oxidative stress normally entails high levels of reactive oxygen species on the seminal plasma. This happens normally when there are infections, when there are problems with the mitochondria from the sperm cells, there is a stressful factor after cryopreserving the sample. Okay, so there are available some chemical tests like this one. Okay, in which you can just quantify colorimetrically the level of ROS on the seminal plasma. Hence, you can just do a fast prediction about if the membranes of your cells could be damaged or not, or which is the risk of DNA fragmentation in your sample analyzed. But when you are detecting high levels of ROS and then you perform the DNA test and also there is a high level of fragmentation on the DNA, what to do, how to handle that samples. If you want to do an ICSI or if you want to do an IVF and you need to use that sample, how to handle. You will find in the market this profile of kits in which you will mix your semen sample, you will incubate it with some magnetic beads that are coated with some proteins that have a high affinity for trapping these sperm cells that are damaged. Hence, you put this mixture in this type of tubes and put it inside a magneto. This magneto will attract all these magnetic beads around the walls for the tube and will leave alone, freely swimming, these sperm cells that are not damaged. Therefore, the technician could easily aspirate that sample and use it for doing the ICSI or the IVF, minimizing the risk of selecting one sperm with DNA damage. On the same way that the reactive oxygen species are affecting on the integrity of the DNA, also it could affect on the integrity of other membranes like the plasmatic membrane. This is why when there is a poor motility or after cryopreservation, it is recommended to do a specific assessment of membrane integrity. Conventionally, it is used the host test, in which the sperm cells are incubated with a hyposmotic solution, and we are evaluating the response of these cells. If the tails are rolled, it means that the integrity of membranes is working properly, and therefore the sperms are good. If the sperms are not responding to the swelling, it means that the membranes are damaged. Okay. Additionally to that method, WHO recommends using the eosine microsheen staining methods. These are very popular. You will find lots of different kits that allow this type of coloration, but we need to take into account that normally these kits results in a very noisy background, and the contrast is not always the same between the dead and alive sperm cells. In some cases, there is an overestimation of dead sperm. In other cases, it's very difficult to find sperm because there is a very noisy background that difficult the analysis. This is why MICROPTIC has developed the Brightbit kit. Okay, here there are the vitality slides. SCA, this is a kit composed by some slides and this reagent for staining the dead and alive sperm. The advantage of that kit is that allows to do a double test, allows to evaluate colorimetrically the dead and alive sperms with having a good contrast and a very clear background, but also this is acting like a host test, is slightly hyposmolar, and therefore it is inducing the swelling or the coiling of the tails or not, depending on the viability of the tails. Okay, so it allows to do a double test at the same time by using the same kit. But on the same way that happens for motility and concentration, for these difficult samples that contain lots of debris, low sperm numbers, and that are very difficult to handle by using this type of bright field methods, you can also use fluorescence methods for evaluating the viability of membranes. MICROPTIC could offer the FluoBit kits. This kit allows to stain on a blue coloration the alive sperm cells, in red the dead sperm cells. Again, it's very important because these dyes will highlight the sperm cells and will omit all the debris, all the noisy background that could difficult the analysis. Okay, so it is recommended for these pathological or very complicated samples. On the same way that the plasma membrane is affected by the method, the protocol that you are using, it could be the freezing or the reactive oxygen species, the acrosomal membranes could be also affected by these processes. So in some cases, laboratories decide to do a specific test for evaluating the sensitivity of acrosomal vesicles to react before an IVF. Okay, this is just to predict if the subpopulation select will be able to penetrate or not the oocyte layers. You can use this type of kit like the FluAcro kit that will entail all these reagents required for doing a specific labeling of the acrosomal vesicle and the postacrosome, okay, and induce or simulate in vitro their acrosome reaction. On the same way that you are using that type of kits, you will see that there are also available other biochemical kits, that could be the AcroScreen, that allow a quantification, colorimetric quantification of the levels of acrosine inside the acrosomal vesicles. Okay, because it's not only important to see if the sperms are reactive or not, also it's important to quantify if the quantity of acrosine released it is enough to make a hole on the zona pellucida. Therefore, this type of test could provide from very valuable information for predicting the success of an IVF. Just to finish with this block, with this chapter, I would like to talk about the round cells. We have to think that when we are evaluating motility and concentration, also we need to take into account if there are other cells that are not sperms within the ejaculate. Okay. In that case, there are different staining kits available in the market that allow to evaluate these round cells. One of the first kits that we need to use are these staining kits specific for labeling peroxidase activity. When there are leukocytes, when there is an infection and these leukocytes are activated, the percentage, the concentration of peroxidase positive leukocytes is very high in the ejaculate. When there are more than 1 million peroxidase positive cells per milliliter, there is a high risk of infection, and this patient should be immediately treated. Okay? This is why this fast test, like could be the leukoscreen, MICROPTIC is offering the leukocyte slides that contains not only the reagents for labeling, also the slides for doing the analysis. Okay, so you can just easily quantify the number of peroxidase cells in the ejaculate and discard the presence of an infection in the patient. But what happens if after doing that type of test there is not labeling, there are not peroxidase cells, but there are so many round cells? WHO recommends to do a specific morphological evaluation of round cells. Here the Papa Nicolau coloration has a critical role because this is the most popular coloration used among laboratories, but it entails a big problem. There is not a standardized protocol. Each laboratory is using its own protocol, and therefore the final concentration, the final coloration obtained could be very different between laboratories. This is why some laboratories prefer using this type of coloration, like the DIF-Quick or the sperm stain ready to use, that are composed by 3 solutions. Okay, that allows not only the differentiation of the anatomy for the sperm cells, but also for these round cells on a fast way, on an easy way. So now that we have talked about the difference between these disposable, it's moment to think about if the results or if the disposables that you are using in your routine are accurate or not. Okay? This is why it's important to take in mind the timing for doing each test. There are some sperm parameters that are very sensitive to the pass of time. This is why the WHO has defined the following timing for doing the test. Okay? So once sample is collected, should be left to liquefied, and during the first 30-60 minutes, you need to do the evaluation of the appearance, the volume, the pH. If you suspect from the presence of ROS, also this fast test for ROS, motility, vitality. You can do the smears for morphology, account for the sperm number. If there are so many round cells, evaluate the level of peroxidase activity on the ejaculate, or if there are agglutinations, evaluate for the presence of antibodies in the sample. Okay, so there are lots of tests to do in this short period of time. This is why it's very important, especially if you're receiving multiple samples at the same time, to prioritize, to do a priority of test and have it clear the timing in order to get realistic results, to not affect the results by the factor of time. Okay? After this first hour, you have time enough for then evaluating the morphology. Okay, do complementary tests, or if you are interested in doing DNA or acrosome reaction, you can also do it as soon as possible, but during the same day. It's not necessary to be done immediately. Okay. So at the same time that you need to think about which is the priority for doing each test, it is important to think about the accuracy of the disposables that you have selected for doing each test. This is why I have included in that presentation multiple papers that are comparing different methods, different disposables for doing the same test. Okay, so I would recommend just to read it after this webinar, and if you have any question or any doubt, just you can contact us. Okay, we will try to solve your doubts. Okay. Now I would like to finish just thinking about how can you improve the efficiency of the procedures performed in your routine. Okay, we have talked about disposables, we have talked about timing, okay, but this is why it's important that when you are doing an analysis manually, there is you can invest a lot of time for doing the test. Not all results are accurate. There is a high inter-technician variation. It is very difficult to learn and be properly trained, and there is a high probability to make mistakes during transference of results. This is why the incorporation of a CASA system in a laboratory could solve all that problems, could help to standardize all the process, improve the time, improve the results, the quality, okay, and maintain all the traceability for doing the analysis. So it will provide from a very huge advantage, especially if you want to be accredited. Okay, here I have summarized in that table just a comparison, a time comparison of the time that is invested by a technician if you're doing a motility and concentration test, vitality, morphology, DNA, and leukocyte test. Okay, so when you are doing manually all these tests, a technician could invest close to 50 minutes for completing the analysis. Conversely, when you are incorporating a CASA system, that could be the SCA, in just 26 minutes, the analysis is done. But if you invest in more advanced devices, like could be the automatic SCA scope, in just 16 minutes, you can finish the full analysis of all these tests, but taking into account that this is an automatic method and it's not required the presence of a technician handling the system during these 16 minutes. Okay, so meanwhile, this technician can be working doing other things in the lab simultaneously. Okay, so here the efficiency of the routine in a laboratory could increase drastically. This is why I would like to finish that webinar just showing some questions that you need to think about concerning about which is the best way to minimize the risk of contamination in your lab. If there is any way to standardize the analysis procedures that you are following, how can you reduce the time per analysis, or how can you reduce the cost of this analysis? So, for example, for minimizing the risk of contamination, you can use a single-use disposables. You can wash the samples in order to minimize the contact with the plasma seminal. Okay. Also, you can mount the smears to minimize the release of aerosols during the analysis. For standardizing, you can just obviously introduce a CASA system, but also you can use this type of disposable that will help to standardize your sample preparation for morphology, for instance. Also, you can use some barcodes that could help to transfer all the information for the sample, for the patient, to your laboratory informatics system. Okay. Also, it's important to take in mind the timing for each analysis in order to reduce the time of analysis. You can pull different samples and do the same test simultaneously for different samples. Also, it's important to think about to have a preference for these disposables that are fast, are quick disposables in which there are does not entail there is a lot of steps. Okay, you can do the test easily and quickly, and obviously CASA system will help you to analyze everything faster. Concerning how to reduce the cost of each analysis, you can select, depending on the number of samples that you are working with, you can just select some disposables with multi-chambers slides, or maybe a reusable counting chamber, or also if you are using staining solution, you can just do some aliquots and preserve the stock solution on the fridge in order just to enlarge a little bit the life period for that solution. Okay, so there are so many points to think about, to discuss with your technicians, to your team, in order just to improve the efficiency for your laboratory. Okay, so thanks for all, thanks for your patience. Now is your turn. You can just ask your questions, and if you have any doubt, you can just contact us by email. Thanks at all.